deoxyribose, right? And that's where the name comes from. The sugar and the phosphate, they act as the backbone of the molecule. They help hold everything together. The bases, the nitrogenous bases, a fancy word there, it only means that it's a base that contains nitrogen, right? So nitrogenous base is going to be one of our four letters, our A's, T's, C's, G's. Derby. Sit down. You don't remember at all that you're not supposed to be there? Izzy. Okay, so the 5-carbon sugar is also known as a pentose, by the way. So like a pentagram is a 5-sided shape, so a pentose is a 5-carbon sugar, the name deoxyribose. So when we look at the actual picture of it, right, we have a sugar, phosphate, sugar, phosphate, sugar, phosphate repeating pattern there on the edges, right? They act as the backbone, the structure that hold the whole thing together, right? The instructions themselves are located in the bases. These are the nitrogenous bases: adenine, thymine, cytosine, and guanine. You do need to know the full name. You can't just know the A, T, C, G. Okay? These are called the nucleotide in one. So one sugar, one phosphate, and one base, whether it's the A, T, C, or G, is known as a nucleotide. This is the monomer, remember, of DNA. We can start sticking these things together, and you're actually going to be modeling this for me, especially next interview when you talk about DNA replication. But how do we actually build new DNA? So you have to know the nucleotide, right? And you're actually going to be building nucleotides for your mural. Okay. The four bases, right? I do need to know the full name of them. Adenine, thymine, cytosine, and guanine. They have a particular sequence in order in which they can go, right? When there is a problem or a mutation, basically what you're having is a change in somehow the sequence of our bases, or A's, T's, C's, and G's. Right? This will create, of course, problems in what type of protein we can create. It's like taking your recipe to make a chocolate cake and then kind of taking out a few steps. Right? If you do that, you're probably going to end up with a pretty bad tasting cake, right? Or something's not going to work quite right. It's nearly the exact same thing for how we make proteins and use the protein, right? So it's not going to work as it should. The adenine and guanine are a 2-ring structure called purines, right? It has this basic structure here. And the cytosine and thymine are a 1-ring structure called pyrimidines. Purines will always connect to pyrimidines, and more specifically, the A's, the adenines, will always connect to the thymines. The guanines will always connect to the cytosines. This is called Chargaff's rule. All right? And you can see that up here in our model. A's connect to T's, C's connect to G's. Doesn't matter the sequence. It always comes to that basic structure. The shape of the DNA molecule is called the double helix. So as we actually put this together and get it off of a 2-dimensional structure, we get a spiral, right? The blue indicating the sugar phosphate backbone, and then the colors across the middle indicating the 4 bases, right? Yeah, this is all very simple stuff, right? And by now there should be a review for you. Chargaff here is, Chargaff's rule is what he's the scientist who figured out that there's always going to be an equal amount of thymine as there will adenine, and there will always be an equal amount of cytosine as there will be guanine. And so he's the one that figured out, oh, well, adenine and guanine, adenine and thymine go together, cytosine and guanine go together, right? And so we call it Chargaff's rule. Also call it complementary base pairing. So I will use that term. But base pairing is just how we connect the bases together. A's to T's, C's to G's. Okay. The other thing about the, am I going to show it here? No. Let me back up here just a second. The other thing about and I'm just going to mention it here. We're going to get into more detail about this later on. About the DNA molecule is that it's a directional molecule. Right? And so we haven't covered this yet, so hopefully you guys are paying attention here. So DNA molecule we know has 2 strands, a double helix. One strand goes one direction, but the other strand actually turns and goes the opposite direction. It's just like a road. Cars can go on one side of the road in one direction. On the other side of the road, they have to go in the other direction. Now, when we're just talking about the structure of DNA, that might not seem important, but when we talk about how we create new DNA, well, it's going to become very important, right? And it's going to make the DNA replication and the creation of DNA kind of a complicated process, right? We determine the direction that the DNA is going by what we call the prime, right? So I follow closely here. Remember, in this sugar here, we'll look at this one. In this sugar, this is the sugar, there are 5 carbons. It's a 5-carbon sugar, right? Each sugar has a number, right? Each carbon has a number in that sugar. The first carbon is the carbon attached to the base. So this is 1, 2, 3, 4, and the 5th carbon is the one attached to the phosphate. Okay, so we call this 5 prime, and we call this basically 3 prime. Okay, and this determines the direction of the molecule. Basically, which one is higher up, right? In this case here on this side, we call this the 5 prime end of the DNA molecule because the 5 prime carbon is pointed upward. The 3 prime carbon is pointed downward. So that means on the opposite side of that same strand would be the 3 prime end. This is the 3 prime carbon right here. This is the 5 prime carbon here. So what we're showing here is that the direction of this molecule is going down, right, or downwards. If you look on the other side, you will notice that it's the opposite, just like a road. The other side, our sugars are flipped the opposite direction. They were essentially pointed in the opposite direction, right? Again, we have our 1 prime, 2 prime, 3 prime, 4 prime, 5 prime sugar. If you were standing on your head, it would look exactly the same as this, right? But then now this would be the 5 prime end of this molecule. This would be the 3 prime end, right? It is what we call anti-parallel, right? It's parallel, it runs right next to each other, but anti-parallel because it goes in opposite directions. Okay, so that term is on your interview sheet. Make sure you understand how to explain it. One end of the molecule is the 5 prime end, one end is the 3 prime end. And of course, the other side would be opposite, right? And we determine that by the sugar that is in the deoxyribose, all right? That is the deoxyribose. Questions about the direction of the DNA molecule and being able to explain that, right? This is really the only complicated part of this. We will actually put it to use when you start building DNA next interview, but right now I just want you to be familiar with the idea of the direction. Okay. So again, kind of an overview. When we're looking at DNA, we're looking at a thing called genes, right? And genes are a specific sequence of DNA that is going to code for a specific trait, right? We often refer to genes for eye color, or genes for how tall you're going to be, or genes for hair color, right? So it could code for a sequence of different proteins, but it's really coding for one particular trait, right? A trait, something that is expressed, something that we actually see, okay? So in the DNA of the cell, or in the chromosome, we have the DNA, and then in the chromosome. In the nucleus, we have the chromosome. The chromosome is composed of DNA, and on the DNA, we can find these genes, right? And what's cool and what we're doing in science right now is we're actually trying to figure out where the genes are, right? And this is what so much money is being put into, because once scientists can figure out how to manipulate genes, how to turn genes on and off, they're going to be able to get rid of disease as we know. It won't exist anymore. You see science fiction movies of people taking a pill and then they don't have cancer anymore, or whatever disease, right? That is actually reality. We're getting closer and closer to that. We're still a long ways off because we don't understand the DNA yet, right? There's so much we don't understand. It's like this endless library of information, and we're only seeing a couple chapters of one book right now, right? But we know as we open up the information, life on this planet as we know it is going to change, okay? So, the last thing that we're going to talk about is this idea of epigenetics, all right? And then we'll be done. Actually, I'll talk about this tomorrow. Let me get into the mural and just kind of show you what you're going to build. Looks like I closed my mural.